Thank you for joining us for the 2021 Phospholipid Division Best Paper Award lecture, Delivering RNA to New Tissues by Testing Thousands of Nanoparticles in Vivo Using DNA Barcodes. First, let me thank the American Alchemy Society for organizing these virtual webinars and many other courses. They're doing a tremendous job. Also, I'd like to thank the International Lecithin and Phospholipid Society for sponsoring this award. And I am Ernesto Hernandez. I am chairman of the Phospholipids Division. And it's my pleasure to, I'm also with Advanced Lipid Consultants, by the way. And it is my pleasure to introduce Dr. James Dahlman as the recipient of the 2021 Phospholipid Division Best Paper Award. James Dahlman is an assistant professor in the Department of Biomedical Engineering at the Georgia Tech and Emory Medical School. His lab works at the interface of drug delivery, genomics, and gene editing by applying technology development approaches to nanomedicine. James' students have developed DNA barcoded nanoparticles to measure how hundreds of nanoparticles deliver messenger RNA and siRNA in multiple cell types in vivo, all from a single animal. Since 2016, the lab has used DNA barcoding to quantify thousands of nanoparticles in vivo, thereby identifying nanoparticles that deliver RNA to new cell types without targeting ligands. His lab uses these approaches to improve cell type specific gene therapy, targeting, and identify genes acting as master regulators of nanoparticles delivery in vivo. He was a co-founder and board member of Guide Therapeutics, which was recently acquired. James has published in Science, Cell, Nature, Nanotech, Biotech, BME, Science, Translational Medicine, and other high-impact journals. Since starting his lab in 2016, James has won the Rita Schaefer Award, the ASGCT Outstanding New Investigator Award, GT Outstanding Achievement in Early Career Research Award, Tech Review TR35, Control Release Society GDGE Award, has had his barcoding work described as a top 10 emerging technology by the World Economic Forum and won numerous Young Investigator Awards. James graduated with a bachelor's in biomedical engineering from Wright State in 2009, received his PhD in 2015 from the Harvard MIT HST program, where he studied with Robert Langer. As a postdoc, he studied CRISPR-Cas9 with Fang Xiang. Please write any questions for our presenter into the question section of this presentation for the control panel. All questions will be addressed at the conclusion of the presentation. Thanks again for joining us. With that, I will turn it over to Dr. Dahlman. Thank you, James.
Well, thank you so much for the invitation. Just confirming you guys can hear me okay, right?
Yes.
Yeah. Great, thank you. Yeah, so I'd first like to thank AOCS for this award. I'm really excited to be here with you guys today to describe our lab's work on DNA barcoding and to go a little bit more in depth into the specific paper that won the award on what we call constrained phospholipids. I think you might find that kind of interesting. And I really appreciate the kind introduction. You know, I think phospholipids is a very cool, I think it's a great community. I think phospholipids are sort of underexplored in my field. I'm hoping that we can build a few bridges between, you know, the biochemists and lipid chemists and folks like me. So thanks for having me. As a disclosure, I have to tell you that I co-founded Guide Therapeutics, which was acquired by Beam Therapeutics, and now I consult for Beam Therapeutics. So I want to start off with one of the more important slides here, which is to describe sort of how a pretty simplified version of how I think about gene therapies work. To be more specific here, when I say gene therapies, I mean non-viral gene therapies. For those of you in the audience who are familiar with gene therapies, you may be aware that you can sort of subdivide gene therapies into those that are based on what are called lentiviruses, those that are based on what are called AAVs, and then those that are based on non-viral gene therapies, usually carried by conjugates or nanoparticles. If you've had either the Moderna vaccine or the Pfizer-BioNTech Acuitas vaccine, you've had a lipid nanoparticle carrying an mRNA injected into your arm. So we're talking about non-viral RNA therapies today, and in particular, we're talking about the subset of those therapies delivered by LNPs, lipid nanoparticles. So today, we're going to be focusing on work that was largely been driven by Kalina, Melissa, Afsane, and Corey. And there's some other members of the lab who contribute as well, but a lot of the data is going to come from these folks, and I just want to shout them out real quick. The other thing I want to shout out is if you think about what gene therapies require, and if you really want to make a gene therapy drug, you need to have intellectual property around that drug. Obviously, you need to be able to manufacture that drug, which can be challenging sometimes. But in addition to those things, I would say you sort of need three things. You need really great payload, meaning if you're going to use an siRNA drug, the quality of your siRNA has to be really, really good. If you're going to have an mRNA drug, the quality of the mRNA itself has to be really, really good. So the payload itself has to be optimized. We don't do that, but it's really important. And also you need a great gene target. So, it's not just delivery. Delivery is necessary, but not sufficient to get a drug across the line. So I just want to really highlight this is a team effort here. We focus on the delivery, but you have to have great folks working on payload and target as well. So very quickly, I just want to highlight Phil Santangelo, who is my payload guy. He makes great mRNA. If you're really interested in how mRNAs are made, synthesized, purified, characterized, and so on, especially now that many of you have been injected with them, I would really encourage you to read Professor Santangelo's work. And then, one of our collaborators on gene targets is a guy named Eric Sorcerer, who's an MD over at Emory. And again, Eric's job when we work together, I do the delivery, his job is to figure out what we should be targeting. So again, this is a team effort. And the thing I want to highlight about genetic targets is the more simple the target, the better. So an ideal target is, protein X causes disease X, and then you inhibit protein X. That's the best case scenario. A worst case scenario is protein X goes to Y, goes to Z, goes to A, goes to B, which goes to C, which causes disease or whatever, some complicated cell signaling diagram. That's going to be harder to make a difference in that situation. So if you have a really simple cell signaling diagram where protein X is causing disease X, that's the type of gene target you want. Okay, so let's get into the delivery stuff. And first, let's talk about why RNA therapies are interesting. First, we have the mRNA vaccines. These things are, I don't use this word lightly or loosely. I consider them to be miraculous drugs with a lot of work put into them beforehand, but they are working better than I ever thought they would, even though I'm an LNP mRNA person. The efficacy of both the Moderna and Pfizer vaccines has been tremendous, over 90%, 95%. These things have held up against continuously changing variants. It's just a fantastic drug, and it was developed, as we all know now, less than 1 calendar year after the sequence was developed. I mean, it's just crazy. And I think that as we move forward, it's clear from recent reports put out by Pfizer and some other big pharma companies that vaccines may be mRNA based for a lot of folks from now on, not just COVID vaccines. So I think we're at the beginning of a very exciting sort of mRNA generation. Another thing that's a little older, but I think it's just as important. And again, for those of you in the RNA field or familiar with the RNA field, you'll be familiar with this already, but I just want to highlight this for those of you who may not be familiar with my field. These are human data. This is New England Journal of Medicine from 2016, it's a while ago. But what you're seeing here is gene silencing in human beings after a single administration of a drug called inclisiran. And whenever you see a drug that has the last 5 letters are S-I-R-A-N, sarin, that is an siRNA drug. Just like if you see a drug that goes mub, if it has mub at the end, it's a monoclonal antibody. Similarly, if you have inclisiran, means it's an siRNA drug. So the thing that's really cool about this is that inclisiran targets a gene called PCSK9, and it turns out if you knock out PCSK9, you dramatically reduce serum lipids, And so what you see here, and think about it, an siRNA is a small RNA that turns off genes. So if the siRNA is working, you should see the gene expression drop, and that's what you see here. 0% would be normal gene expression, and what you're seeing is about 75% gene silencing. So 75% less gene expression in these treated patients. The thing that stunned the field and was the fact that you were getting, robust gene silencing for 6 months after one administration. So what this means is if you have high lipid levels, instead of having to take your Lipitor every day, you might be able to get one single subcutaneous, so basically like an insulin shot, one single subcutaneous shot every 6 months. And the fact that you can, take this drug once every 6 months and it's gonna, really lower your lipid levels led to the acquisition of a company called Medicines Company. Medicines Company owned a portion of the inclisiran program. And as a result, Novartis bought Medicines Company for $9.7 billion just to get a piece of the inclisiran program. So again, this is an siRNA-based drug. These results are phenomenal. They've been recapitulated with other siRNAs targeting other genes. But this sort of the era of gene silencing, at least in the liver, because you can deliver stuff in the siRNAs in the liver, is here. So that brings us to what I think is sort of the overarching problem or opportunity, I guess, if you want to call it that in my field, which is the non-liver RNA delivery. So if you think about what's been done in the clinic so far, you have the intramuscular injections of mRNA vaccines, which we're familiar with, but we also have either intravenous or subcutaneous injections of RNA drugs that target the liver for different genetic diseases. Just Alnylam right now, one company, has 13 different programs targeting liver drugs, or liver diseases, 13 programs. If you sort of sum up the amount of money or value, what we call market cap, in just the liver for RNA drugs, is probably on the order of $30+ billion. If you sum up the RNA market cap and all the programs and stuff outside of the liver, and you exclude the vaccines, it's basically zero. So there's a huge problem in that we can't deliver RNA outside the liver right now. But clinicians and even investors have seen that if you can deliver RNA, like in the liver, you can make a big difference in a lot of patients' lives. So the key question in the field is, we have all these RNA payloads that are good to go, they're safe, they're effective, the siRNAs are great, the mRNAs are getting better. How do we deliver them outside of the liver? And I've been a non-liver RNA delivery person since the first time I picked up a pipette as a first-year PhD student in 2009. That's all I've ever done is non-liver RNA delivery. So if you want to go outside the liver, and you know that the liver is really, really good, liver delivery is already really good, it's really helpful to ask a question, well, why has liver delivery worked? Maybe you can learn from that and apply that to other tissues. And one answer, to sum up a lot of literature really, really succinctly, is that the liver structure itself, the anatomy of the liver, the physiology of the liver, actually promotes delivery. If you inject something into your blood, the liver clears a lot of it. And there are physical reasons for this, there are physiological reasons for this. Tons of papers on this. This is one of the many papers I enjoy. I would really recommend you read this if you haven't. It's a great paper. But the second reason I think that is maybe less appreciated within the field, because a lot of people in my field know that the liver is good at picking stuff up out of the blood. It's a lot of folks know about fact number one. What's less appreciated is the fact that there was an easy assay to look for liver siRNA delivery that propelled the field forward. So around 2006, there's a big paper that came out in Nature Biotech out of Alnylam. In part of that paper, they reported what they called the factor 7 assay. And the factor 7 assay was, again, to make a very long story short, a very easy assay to look at how well your delivery vehicle delivered siRNA in an animal, in a mouse, in a living mouse. But it was you could only read out liver delivery. So what that meant was, let's say I was a delivery person. I wanted to make, a nanoparticle delivery system for siRNA, and I didn't know which nanoparticle was going to work. I can make a big particle or a small particle or a charged particle or a neutral particle or whatever I want. I can make thousands and thousands, actually tens of thousands of nanoparticles, but I don't know which one's going to work. Well, if that particle is supposed to target the liver, I can actually use the factor 7 assay and test a few dozen particles in mice pretty easily. You take 3 mice a group, you inject them, 3 days later, you just take a little bit of blood from the tail, the mouse doesn't even have to get killed, and then you basically run a colorimetric assay, it takes 10 minutes, and you can read out delivery, but again, only to the liver. So what this meant, and by contrast, if you wanted to look at delivery outside of the liver, it was really, really hard. I can tell you this because this is what I did during my entire PhD, non-liver. So it was harder for non-liver people to assay whether their delivery vehicles worked, and as a result, the field focused on liver delivery. So you had really big academic labs and really big companies starting to sort of ping pong off one another using chemistry and results that were coming out. And as a result, over 8 years, from 2006 to 2014, the dose required for liver siRNA delivery dropped by 10,000 fold in 8 years. So delivery got at least 10,000 fold better in 8 years because you had an assay that you could use to optimize it. So when I look at the history of RNA drugs, I take of course a look at the liver and hepatocyte targeting RNA drugs, and I think this is the conclusion I would draw. I'd say by iteratively quantifying how hundreds of different nanoparticles delivered RNA to hepatocytes in vivo, in adult animals, in vivo means in adult animals, the field found clinically relevant delivery to hepatocytes. So if you look for hepatocyte delivery, you're going to find hepatocyte delivery. So the operating hypothesis that my lab has worked on and guide was based on before it was bought was that if you iteratively quantify how thousands of nanoparticles deliver RNA to any combination of cell types in vivo, you may find clinically relevant delivery to those other cell types. So I would ask this, I'd say this another way, is this an assay problem? And I would say, yes, it is. So the way that we test thousands and thousands and thousands of particles in vivo, which hasn't been done before in the field, is we use DNA barcodes to do this. And this allows you to test everything directly in vivo. Right now it means mice, but we're developing systems that could be used for other animals as well, larger animals. And it also means that the assays are really high throughput. So right now we can do hundreds of LMPs per mouse. There's some other traits here, which we'll get to a little bit later, which is the assays are functional and they're iterative, but again, we'll talk about those traits a little bit later. Right now, we're going to talk about barcoding, the fact it's in vivo and the fact that it's high throughput. So the way that this works, this is an oversimplified schematic. I show this one not because of its accuracy, but because, like many of you, I'm a nerd, and I've been subscribing to Scientific American since I was like 15 years old. And they asked me actually to write an article about this 2 years ago, so I like to show this because I think it's cool. So, and also it's a nice diagram. It's oversimplified, but it's a nice diagram. So this is how barcoding works from a simplified perspective. Nanoparticle A with chemistry A, in this case, pink chemistry, whatever that is formulated so it carries DNA barcode A. Nanoparticle B with chemistry B, in this case, blue chemistry, is formulated to carry barcode B, and so on and so forth. You don't put, you then pool all the particles together, you administer them to the animal, and then you isolate whatever tissues you want. So in this case, you take out lungs, liver, and heart, and then you say, okay, in the lungs, we get a bunch of orange barcode. That means that nanoparticle C, which was carrying the orange barcode, is better at delivering the barcodes than nanoparticle A or B. So these assays give you basically pie charts where every slice of the pie is a different barcode. If that makes sense. So if the lungs were a pie chart, it would basically be a big orange pie chart with a little slice of pink in there. In reality, there are a few things that we do that are different than this. Number one, it's not just a barcode, there's actually a functional molecule in there, which we'll talk about a little bit later. Number two, you don't put all the particles in. There's this big quality control step where you throw particles out before mixing them together. So in reality, you don't make 3 particles, you might make 200 in a day, and maybe 15 of them aren't any good, so you throw them out, and then you pool the good 185 together and you administer those. Number 3, right now it's mice, but again, it'll eventually be other animals. Number 4, we don't actually do delivery readouts at the tissue level. We don't do lungs, liver, and heart. We actually take the lungs out and we separate them into different cell types. So we have lung endothelial cells versus lung macrophages versus lung whatever. And then we have liver hepatocytes versus liver endothelial cells versus liver Kupffer cells versus whatever. So you actually get readouts not at the tissue level, like it's depicted here, but instead at the cellular level. So these are what the data sets actually look like. I have to actually show this in the field because people don't quite, it's a bit of a, change. Every row here is a particle. Every row here is a particle, and every column here is a tissue or cell type. So just think about how many in vivo data points you're looking at in this little square, right? In this case, we turned it into a heat map because the Excel sheet is basically too big. So can't make bar charts with this. You have to turn it into a heat map. Blue is good, red is bad, but it doesn't matter. This is the scale of delivery. And so you can imagine what this sort of opens up. It means you can do all sorts of structure function analysis and, structure activity relationships, all sorts of stuff. But it's a really large scale of data generation. And as was mentioned in the introduction, I think since we started the lab, my lab has tested probably 5,000 or 6,000 LNPs in vivo. And the company before it was acquired, it actually did far more than that in a very short period of time. The other thing I want to highlight here is that these data, include a bunch of really important controls. In this case, one of the controls is called naked barcode. I apologize, there's no y-axis here, it got cut off. This is basically a percentage on the y-axis. So imagine like you're looking at a pie chart and you said, okay, this top particle, this like top dot is particle whatever, maybe it's particle 35, I don't know, particle 77, whatever. That particle has about 5 or 6% of the entire pie is that particle. Okay, so normalized delivery is just essentially how much of the pie is from each particle. So if you sum up all the particles, you get to 100% of the pie. All right. If you put in a naked barcode, in other words, a barcode that isn't being delivered by a particle, we know that DNA barcodes don't, and DNA in general doesn't enter cells on its own very easily. So the DNA should be sort of the worst, should be delivered less than the DNA within the barcode, within the particles. And that's what you see, all the naked barcodes fall out. The other control that we use is that we quality control the LNP. So like I said, we don't pool all the particles and inject them, you only pool the good particles. So the way that you do this is you make, again, let's say 200 particles in a day or 150 particles in a day or whatever. You then analyze the size and stability of all 150 particles individually. And the ones that aren't good, you throw out. The ones that are good, you keep. And you pool them together. As one of the controls, we then measure. So what you see here on the y-axis is the hydrodynamic diameter measured by DLS for about 100 or so particles. So every dot here is a particle. We had one particle that we made that was pretty small, maybe 30 nanometers, and we had another particle that we made separate chemistry that got up pretty big, 170 nanometers, and everything's sort of in the middle. So after we pool the stuff together, we measure the diameter of the pool to make sure that the diameter of the pool is within the range of the particles comprising the pool. So that's another quality control that we include. We published on this extensively. So, if you're interested in the nitty-gritty details, they're actually kind of interesting. But I kind of want to move on a little bit. So I just want to say that, we can barcode different lipid nanoparticles. So now I'll talk about why this is useful. And we'll start getting into the phospholipid stuff here in a minute. And the phospholipid stuff is related to this. To make a very long story short, the particles that you guys have been injected with, as well as the other FDA-approved particles, as well as FDA-approved particles for siRNA delivery, they all typically have 4 components in them. You have a cationic or ionizable lipid, what I call the designer lipid. That's sort of this blue box. It can be whatever. You can do, I've seen literally, probably at least 20,000, I don't know, compounds made already, and you could imagine up to 200,000 different compounds in this sort of blue category. In addition to that, you add cholesterol into the particle. And, usually people use normal unmodified cholesterol, but there are all these cholesterol variants that actually affect delivery. So again, you could sort of pick from about 100 of those. Then you have what we call a PEG lipid, which has PEG polyethylene glycol on one end, and then a linker, and then a lipid. The way this works in the LNP is that the PEG is hydrophilic. It hangs out outside the particle, because remember the particle is lipid. So the PEG hangs outside the particle, creates sort of a water shield by interacting with water within the blood. And because a hydrophilic PEG normally would not interact with a lipid, you put a lipid on the PEG to sort of anchor it into the LNP. So you can change the structure of this lipid within the PEG lipid, you can change the structure of the linker, or you can change the size of the PEG. And finally, you can add what we call helper lipids or phospholipids all the FDA and EUA approved lipids are DSPC right now, but there are all these variants as well that you can stick in. So you have a 4-dimensional chemical space between blue, green, red, and gray, and each one of these variables matter. And so what you get into is essentially an infinite chemical space. And that's awfully hard to probe an infinite chemical space if you can only test one particle at a time. Second, and this used to be a bit controversial, but it's, I think, not as controversial anymore, is, historically, if you wanted to look for delivery, again, to the liver, you can use the factor 7 assay. Or now for mRNA, it's called the EPO assay, but you can use these assays. If you want to do delivery outside the liver, you can do some luciferase stuff, but the resolution, frankly, isn't great. You can't flow it. You just have to look at tissue level stuff. And it's expensive to do, 1 by 1 by 1 by 1. If you have 5,000 particles you want to test, that's a 15,000 to 20,000 mouse experiment. So it's not really feasible. So what people do is if they want to find delivery to the heart or lungs, they don't order up a 15,000 mouse experiment. They do all the studies in cell culture. So if you want to find delivery to the lungs, for example, you might take A549 cells, which is a lung cell type, stick them in a dish, take all 5,000 of your particles, spray 1 by 1 by 1 by 1 by 1 into the dish. Say, okay, of these 5,000 particles, these 3 particles worked pretty well in the dish in cell culture. So I'm going to take those 3 particles and put them into the animal in vivo. So what you're really saying, if you're doing that, and this is what I did during my PhD, by the way, is if you're going from a big library of compounds that you're testing in cell culture in vitro, and then you're taking a very, very, very small percentage of that, less than 1% usually, and putting that in an animal, then what you're saying is that the cell culture delivery is the best way to predict delivery inside the animal. So, and this is common throughout the field, not just the stuff I used to do. A lot of folks do in vitro, in vitro, in vitro, in vitro, in vivo. That's the workflow. Cell culture, cell culture, cell culture, cell culture, animal. So we asked the question, does in vitro delivery predict in vivo delivery? We took the same pool of particles, we on the same day barcoded them, put them either into cells, cell lines, primary cells, or into animals, and isolated the same cell types out of those animals using what's called FACS. And what you see here is a control experiment where delivery in cell culture in vitro at 1 dose, which is the x-axis, IMAIC is the type of cell that we use, don't worry about it, predicts delivery in the same cell line at a slightly higher dose, which is what you would expect, and the naked barcode, the negative control falls out. So we can say from these data that cell culture delivery, in vitro delivery, predicts in vitro delivery. So once we had that control result, we went in and compared in vitro delivery to in vivo delivery. These cell types are matched. We did this for a bunch of different cell types. And we've never seen in any of our studies any kind of relationship at all between systemic delivery, I'm injecting the bloodstream, in an animal, and cell culture delivery, ever. And I think that the implications of that for the field are actually kind of stark, but I don't want to go down that road at this point. So based on those results, we no longer perform in vitro cell culture delivery studies in the lab unless we're asked to by a reviewer, because we found that basically they don't predict anything in the animal ever. Okay, so we can do high throughput, assays. They're in mice. Let's talk now about the fact that they're functional. This is really important. When I say functional, here's what I mean. Imagine this top row. This is not functional. This would be distribution. This would be where does the particle go? So let's walk through this simple example. Particle 1 gets to the cell, but gets stuck on the outside. Particle 2 gets into the cell, but gets stuck in the endosome. Particle 3 gets out of the endosome, releases the cargo into the cytoplasm. If you read out where these particles are going, all 3 particles look the same to the assay. But in reality, you really want to avoid particle 2, because if you think about how we do this in the real world, you run an assay and you get all these data back and you say, okay, well, based on these data, these chemical structures seem to work and those chemical structures didn't work. So we should, in our next library, we should learn from that. And if you're going to do that sort of iterative analysis, you really want to avoid particle 2. Particle 2 is the worst particle for you because particle 2 interacts with the cell, gets all that cytotoxicity because it's interacting with the cell, but doesn't release. So I don't want to be looking at particle 2 as I'm doing my iterative studies. I want to avoid particle 2. Particle 2 is a very bad false positive. What you want instead is an assay, a functional assay like the bottom row here, where particle 1 gets to the cell, doesn't get in, so you don't read it out. Particle 2 gets into the cell, gets stuck, don't read it out. Particle 3 releases the payload into the cytoplasm. The payload functions. So for an mRNA, that means it produces protein that works. For an siRNA, it turns off protein, and you read out only particle 3. So we have a bunch of functional assays. The one I'm going to talk about briefly is called FIND, Fast Identification Nanoparticle Delivery. And this was reported by Corey Sago in 2018. Corey was a co-founder with me of Guide. This is a workhorse assay over at Guide. We know this stuff really works. It's pretty exciting. So the way that FIND works is you take nanoparticle 1 with chemistry 1 and you formulate it to carry with barcode 1 and mRNA that encodes this recombinase called CRE. And we'll talk about what CRE is in a moment. And then separately, you take nanoparticle in, chemistry in, you formulate to carry DNA barcode in, and the same CRE mRNA. So then you have these particles that are carrying, different barcodes and CRE mRNA, and you put these into mice, which are called CRE reporter mice. Biologists use these mice all the time. They can be what they're called AI9 mice or AI14 mice or whatever. But if you Google the original AI9 mouse citation, it's 1,500 citations or something crazy. Biologists use it all the time. So the reason why biologists use it all the time, and the reason why we chose it is because it's a great system. The way that the CRE recombinase system works is all the cells in the DNA, all the cells have this in their DNA, what's called LOX, stop LOX, and then TD tomato. This is a DNA sequence. A LOX is just a particular DNA sequence, 34 base pairs, but that doesn't matter. It's just a particular DNA sequence. There's a stop construct here, LOX, and because the TD tomato is in front of the stop, under normal conditions, the TD tomato is not turned on. But CRE is this bacterial recombinase that is designed to cut whatever is inside a LOX construct. So if you had LOX, stop LOX, TD tomato, if CRE mRNA is translated into functional CRE protein, it's going to cut the stop, which means TD tomato is going to turn on. So if you put this into AI14 mice, only if CRE mRNA is translated into CRE protein and then goes and cuts the stop and edits the genome to cut out the stop, only in that case, so in other words, you have functional delivery, will the cells become TD tomato positive. And then you can fax those cells. And again, so now you have TD tomato positive lung endothelial cells, TD tomato positive lung macrophages, TD tomato positive spleen B cell, whatever you want. And then you just sequence all those different tubes, and now you have a pretty cool readout. So we use the FIND assay all the time. This is one example. We said, hey, people normally use regular cholesterol in their LNPs. I wonder if changing the structure of the cholesterol could affect delivery. And so what we did is we did a really big assay. We did 75 or 80 LNPs all at once in one group of animals, basically, and we varied the cholesterol. And what you see here is a heat map where blue is higher normalized delivery, so bigger slices. The darker blue is basically the bigger the slice of the pie, and the more purple it is, the smaller the slice of the pie. And what we saw is that all the cholesterols where you have modifications that quote the top of the cholesterol, you saw that delivery was better than cholesterols that were modified near the bottom and better than normal cholesterol. So based on that, this is a pooled big barcoding assay. This is, we don't take these data as gospel. We say, okay, based on these data, we can hypothesize that 20 alpha hydroxy cholesterol, the thing you see on the left here, might be better than regular cholesterol. So that's a hypothesis generated by these big screening data. Now you go in and you try to test that hypothesis with individual particles. And that's how, if you read our papers, that's how all of it goes. It goes, we've made a bunch of stuff. We use this cool barcoding thing. The barcoding thing says this might be better than that. And then we validate that hypothesis using individual particles. That's the flow of our papers. So again, I'm skipping over, this is a big advanced materials paper. I'm skipping it, I'm skipping through in like 1 or 2 slides. To make a long story short, we took an LNP, we formulated it with 20 alpha hydroxy cholesterol. Then we took the same LNP and we formulated it with normal cholesterol. Everything else stayed the same. And then we put them into separate mice and we measured the functional mRNA delivery at a really low dose at 0.05 megs per kg, which for those of you in the delivery field of nucleic acids, you know that that's a very low dose. And what we saw is that just like the screen, this is really important, just like the screen, I'm going to go back, the screen predicted 20-alpha hydroxy is better than normal cholesterol. When you tested individually, the 20-alpha hydroxy was better than normal cholesterol. And this wasn't just for Kupffer cells, this was for all sorts of cell types. My terrier is going to come up behind me. He's sweetie. All right, so we use find all the time to do all sorts of stuff. We're going to talk about the phospholipid stuff in just a second. But I also want to talk about one other unpublished paper that we have under review now at I think a nice journal, where find was used to test 144 different LNPs, again, all in vivo across 20 cell types. So think about that, it still blows my mind. 130 LNPs tested in animals across 20 different cell types. So what you see here is we did a bunch of different libraries, and to make a long story short, the libraries are sort of subdivided into 3 groups, which you can think of roughly 3 groups, which we characterize, again, it's cationic, neutral, anionic, but for your intents purposes, just know that the pink LNPs are different from the blue, which are different from the dark blue purple. And what you see is across all these different cell types, by making very subtle changes to the LNP structure, you see dramatic differences. In the pink LNPs, you get really, really good lung delivery. And in the liver, you get delivery that's okay. So if you're looking at the pink bars, you see great lung delivery, liver delivery is fine. If you look at the blue and purple bars, you see no lung delivery at all. And then you see excellent liver delivery by making very subtle tweaks. You can then go in and we say, okay, well, the find assay predicted that this LNP whatever, I don't remember what number it was, maybe it was 35, I think, should be really good in the lung. And sure enough, when we put that LNP into the lung, you get incredible delivery. This is, again, TD tomato positive cells, so the cells that have been transfected with Cree. Almost every endothelial cell in the lung that we pulled out is positive. All right, and then when you go in a little bit farther and say, okay, well, does that new LNP that we just discovered deliver Cree mRNA in the image of the lung? The answer is yes. And then we've subsequently found that LNP delivers Cas9 mRNA and guide into wild-type mice and can preferentially edit genes in the lung without editing genes in the liver. So again, what you see, if I go back a little bit, is you do this big old library all at once in animals. The resulting data give you some hypotheses to test. It says, particle X should be good in this tissue, and then you can confirm it, you can image it, and then you can edit with it. All right, so it circumvents the need to do any of these in vitro assays because you can just do everything right in the animal. So now let's talk about the paper that was awarded. And again, I really appreciate this award. The title is Nanoparticles Containing Constrained Phospholipids Deliver mRNA to Liver Immune Cells Without Targeting Ligands. And I want to highlight Zubao, Melissa, and everybody for contributing to this in a meaningful way. So again, let's walk through what we do. Well, the first thing that we did is we created a big library of new compounds. In this case, we took traditional looking phospholipids and we added all sorts of things to them. And the thing that's most important about this, in my opinion, is at the lower right-hand corner of this phospholipid, you're gonna see an adamantyl group, which is that little kind of hat-looking, diamondy looking thing. Adamantyl groups are lipids that are constrained. So instead of having a lipid tail that's just flopping around, an adamantyl is just folded in on itself, basically. And these adamantyl groups are used in small molecule by medicinal chemists all the time in small molecule drugs. So they have all sorts of crazy traits to them. But what we saw is, I love looking at different phospholipids. Everybody uses DSPC, but there's all sorts of stuff you can try. I haven't seen anything with a constrained group in the phospholipid, at least in my field. And so we made a big library of constrained phospholipids, right? And then we tested them all in vivo. So, first I should say, since I want to go into this paper more in depth a little bit, is we characterized all the phospholipids, and it turns out that these constrained phospholipids form stable particles. So it doesn't really matter. I'm going to go back here. You can see the R group here is basically a free-floating tail that has a length between 9 and 17, and then 17 to Z is slightly different. But all these things have the constrained group at the bottom, the atomantal group. So what you see here is, we actually wanted to know if these constrained phospholipids would formulate LNPs, like stable LNPs, and long story short, yes, they did. It didn't matter if we were formulating them with different lipid lengths or different tail lengths, which is what you see here, A9 through A17. The LNPs have a diameter about 100 nanometers, which is good. They're super stable. So we can formulate particles with constrained phospholipids. In addition, it doesn't matter if you're formulating those LNPs with different cholesterols. So, the constrained phospholipids are good neighbors with cholesterols, cholesterol, and then also modified cholesterols. So these actually form, good particles, which wasn't a given. We then went into the in vivo data and looked to see how nanoparticle structure influenced delivery in general. And what you're seeing here plotted on the x-axis is what we call enrichment. And I don't have time to get into details of enrichment, but basically it's a way of saying, okay, if the lipid structure didn't matter, the enrichment for everything would be 0. If the lipid structure does matter, and as a result, this lipid is overrepresented in good particles that deliver efficiently, and is underrepresented in particles that do poorly, you're going to see a positive enrichment, the green bars. By contrast, if a lipid is overrepresented in particles that do poorly and underrepresented in winning particles, the enrichment is going to be negative, which is the gray bars. So you can see that in particular, when I look at this, I say, oh, that's interesting, kind of carbon lengths of, between, 11 to 13 look pretty interesting, right? So again, this isn't gospel. This is not saying this is exactly the structure activity relationship, but it gives us something to study and a hypothesis to test. So anyway, based on all this stuff, we decided to focus on a particular lipid, which you see there on the left. And what we saw is that lipid is able to deliver RNA to a bunch of different cell types within the liver immune environment, more so than hepatocytes. So you get better Kupffer cell, which is macrophage delivery, than hepatocytes. You get better endothelial cell delivery than hepatocytes. And you actually got some smatterings of lung delivery, although it's pretty low. All right, and then when we go, these are the screening data. When you go actually test, the individual winning compound, you see the same thing. So again, the screen predicts the screen tends to predict the individual compound. So I'm running up on time a little bit, but the last thing I want to do is just highlight that we have assays that are different than FIND. So the FIND assay we've been talking about is the Cree mRNA-based assay. To make a very long story short, we have found that mRNA delivery does not predict siRNA delivery, and siRNA delivery does not predict mRNA delivery, which means that if you're looking for a delivery vehicle for mRNA, you have to use an mRNA functional screen like FIND. If you are looking for siRNA delivery, you have to have a functional siRNA screen. This is that screen. The way it works is nanoparticle 1 with, gray chemistry 1 gets barcode 1 and siRNA targeting GFP. In other words, siGFP. So this will turn off GFP if it works. Nanoparticle 2 with chemistry 2 gets barcode 2 and siGFP and so on. Quality control everything, so not everything goes in. Put it into GFP mice, so all the cells in the mice express GFP. If the siRNA does its job, it's going to silence GFP. So in this little schematic, where the siRNA actually functions, because it's a gene silencing thing, it would be functional in these white cells, these cells that aren't green anymore, because the si turned off the GFP. So then you fact sort the GFP low cells, the cells where the GFP expression is low, and you sequence. So sort of like the reverse of FIND. We use constrained lipids in this. There are constrained ionizable lipids in this case. The assay said, hey, you're getting gene silencing in splenic T cells, which is really, really interesting to us because these compounds did not have targeting ligands on them. So it does not have some sort of CD4 targeting ligand on it. It's just an LNP, just lipids. And hey, by the way, you're not getting liver silencing. So not only are you getting T cell silencing, but you're not getting liver silencing. When we go in and do the actual assay and confirm the winning particle, you see the same thing. You see a dose response with SIGFP and T cells. You don't see silencing in hepatocytes. So what we have is a T cell targeting vehicle without ligands identified in a new chemical space in one go with a single screen. I love this compound. Would have never thought of this before if we hadn't done the screen. And finally, something I think it's a little bit more intuitive. These assays are iterative. And so remember, these are very, very large assays. And so what you can do then is you can take a starting LNP with a starting chemistry, you can test a bunch of variants of it, identify cells that you want to target. We have a bioinformatics pipeline that's on our website to analyze, go straight from barcoding data to analyze how in nanoparticle size or PEG or whatever, all these chemical traits affect delivery. So, you get this giant data set and you say, okay, well, what promotes delivery? Is it the particle size? Is it the PEG? Is it the cholesterol? Is it the whatever? And then use all those data to refine the chemical space, and you do this iteratively. So this is how we quickly improve delivery. And we actually use this to evolve an LNP that targets the bone marrow, which for me has been a bit of a white whale. I've never been able to target the bone marrow. And actually a year or so later, some folks at MIT recapitulated these results with an LNP that was very, very similar to ours. So we've seen this in other people's hands as well. Last thing I'll say here is, we talk about barcoding a lot and all these things. That I consider barcoding to be something out of a technology development lab. So, as mentioned, I did my PhD with Professor Langer, and there I was a delivery person, and then I did my postdoc with Professor Zhang, who's a CRISPR person. And of course, I learned how to gene edit, but in Feng's lab. But more importantly, I was exposed to these like big data ideas that I wasn't very familiar with, and these technology development principles, these technology development labs. And these technology development labs usually create this like amazing technology to read something out, and then they apply it to immunology or to neuroscience or to vascular biology or to whatever, really awesome stuff. But I was like, man, we should be applying these same approaches to delivery, because delivery is a very interesting phenotype. You put something in the body, it has to go here, but not there. And so I have a vision for how technology development can be applied to nanoparticles and nanomedicine. It's not the only vision, it's just a vision. This is a vision, so if you're interested in that, take a look. And I will tell you that over the next year or so, we're going to be publishing entirely new approaches that are different and distinct from these barcoding things. I think they're pretty exciting. There's a big space, is what I'm trying to say, between technology development and nanomedicine. So, yeah. So with that, I want to thank collaborators, funders, and trainees. At the top, you're going to see collaborators and the grants that their PI's on, where I'm kind of hanging on, And then at the bottom, you'll see key members of the lab, the rest of the lab here on the right, and then PI, and then grants where I'm the PI on those grants. So thank you to everybody. I'll leave this with the summary very quickly. Most importantly, cell culture delivery does not predict systemic delivery in an animal. Kind of sucks, but it's the reality. I think if you iteratively quantify how thousands of particles deliver RNA in vivo, you may find delivery to different cell types. And then what we found over and over is that very small changes to the LNP, making 20 alpha hydroxy cholesterol instead of cholesterol, can alter delivery in vivo. So those are things I really want to drive home. And the last thing I'll say, since we have 10 minutes, is I am currently looking for lipid chemists and lipid biochemists. So if you know folks that are interested in doing, pretty work that's pretty out there, work that has been translated effectively into a company. We have a second company brewing the lab right now. We are being tasked with making all sorts of vaccines for people. We're doing all sorts of in vivo gene editing stuff. We have this whole suite of new technologies that we're going to be publishing. It's going to be a very exciting few years. We need 2 or 3 people who have lipid chemistry or biochemistry experience. So if that's you, or that's one of your colleagues or one of your friends, have them email me, because we're going to be hiring people in the next, few weeks to months. Okay, and we're looking at technicians, PhD students, postdocs, whatever. We were very fortunate to get a lot of new funding recently, and so we're going to be growing the lab. So with that, I will stop the share and take any questions. So thanks very much for your time.
Thank you, Dr. Delman. That was very, to say the least, very interesting. This is definitely at the forefront of lipid and phospholipid research, and we are going to see the results through the delivery of vaccines. Specifically, you mentioned the Moderna and the Pfizer vaccines being the ingredients being lipids, or some of the ingredients being lipids and phospholipids, to be able to cross the membrane phospholipid bilayer, and then the organelle membranes and so on. So this nanoparticle technology is definitely nothing short of revolutionary. So thank you very much. We really appreciate your presentation.
Thank you.
I have not seen any questions yet from our attendees. We did get quite a few attendees. But I have a quick question, if I may. It has to be asked, I think. Considering that this, new science, literally, is so revolutionary and so transcendental in the delivery of drugs and messenger RNA and siRNA, how are the regulatory oversight agencies keeping up with this?
Yeah, so I think luckily for all of us, I think although the mRNA vaccines were, put out quickly, there was a bunch of science behind it. So, there's an FDA-approved lipid nanoparticle that delivers siRNA. It's called patisiran. Well, the formulation is called patisiran. That went through the normal, very long, lengthy, expensive FDA approval a few years ago. Moderna's actually been doing, chikungunya vaccines and other vaccines with mRNA for a long time. So all that stuff was sort of in place, ready to roll when COVID hit, which is, very fortunate. And then BioNTech's also the same way. I mean, they didn't develop their own LNP, they got theirs from Acuitas. And Acuitas's lipid has been well characterized for a long time. So, I think moving forward, I think moving forward it's going to be more traditional. I think moving forward you're going to have, years and years and years and years of like painful, expensive stuff, and every new lipid that's put in, or every new lipid formulation that's put in, is going to go through tons of stuff. We were just very fortunate that in this case that the formulations were already set, and of course, as you probably know, they didn't do everything in series. They basically did everything in parallel at great financial risk, and it happened, to pay off. So I think it's going to be pretty normal in the future. Some stuff will be tolerated, some stuff won't. The stuff that isn't won't pass, and the stuff that is will.
That is great. And our, the safety safeguards are there to,
Oh yeah, for sure.
to make sure that
think about the size of the phase 3, right? The phase 3 trials for each of those things was 30,000 people each, and also just think about, we saw this with the other vaccines, the adenovirus vaccines. There's so much scrutiny on these things that if you had any safety issues at all, it was going to be a headline story. If you think about the issues that were coming up with the rare blood clotting issues, think about how much press that got. You have an adverse event less than 1 in a million times. It turns out if you look at the odds of just getting those same blood clots, there's no difference between the treated group and folks who were just sitting around. But it still made, front page news for weeks. The scrutiny is unbelievable. So I think it's the fact that scrutiny is that high and that these things have been cleaned this long is unbelievable. It's crazy to me. That just shows you how effective and how safe they are.
That's good to hear. That's good to hear. We still have some attendees, but I have not seen any questions from anybody.
It does look like we have one.
Okay, I see one here. Hi, great presentation, Professor James. A concern where can one find the bioinformatics follow-up steps that were in your slides? Just a curious bioinformatician.
Yeah. By the way, I'll say for the record that I'm not a good bioinformatician, so it might not be great, but I can tell you where it is. I just, I trust my students to, like, they say it's ready to go, I say okay. So I'm not a coder, never have been. But it's on our website. We have a resources tab on our website, and the current bioinformatics stuff is, I think, supposed to be at least pretty simple. Like I said, in the next year or so, we're going to have a much more advanced technology where we actually had to do a bunch of coding, and that'll also be out in about a year's time. But it's on the website, and there's some, I think there's some GitHub or something associated with it. So yeah.
That's great. That's great. Yes, and AOCS has your contact information if anybody wants to follow up, and especially for the personnel that you are looking for. I don't see any more questions. I do have one that also has to be asked. I think I guess obvious question would be how hopeful is this new delivery science is for finding cures for cancer, for example?
Yeah, I think cancer is going to be tough, but I think that although it's funny, before BioNTech got into COVID, their claim to fame was developing what they call personalized cancer vaccines using RNA. So the way these work, they had a Nature paper on this in 2016. The way these work is, they would take a melanoma. It was melanoma at that time. Take a melanoma, sequence it, take blood cells out of, take cells out of your blood as control, sequence those, compare the sequencing, say, okay, here are the mutations, and then, let's make a vaccine against those specific mutations for that individual person. And they showed those results, and they were phenomenal results. I expect that, I don't know what's going on inside those companies, but I would imagine that after, I imagine they're going to have COVID vaccines being developed from here on out. But in addition to that, once COVID sort of becomes just, it's just going to be endemic. It's going to be like something that we fight with, like the flu from now on. They'll probably expand into cancer vaccines. When it comes to, just traditional RNA delivery to tumors, that's really, really hard to do. But there are other cell and tissue types that I think are going to be a little easier, immune cells, HSCs, so on, where you could, envision, especially if you're delivering a nuclease, like a CRISPR construct, you could envision, treatments that go and try to, cure beta thalassemia, or go and try to cure sickle cell. So there's going to be a lot of advances. I think the next wave of advances outside the liver is probably going to be within the immune system. And then after that, hopefully we can get into, some other tissues as well, maybe the lung or so on, but that's my guess.
That sounds great. That sounds very good. Well, I don't see any more questions being posted. So thank you so much. That was fascinating, interesting, important. Amy, are you there? Can you send us off?
I sure can. Congratulations, Dr. Dahlman, and thank you, Ernesto, as always, for being a wonderful moderator and AOCS member.
You're very welcome. Thank you. Thanks.
Thank you. Appreciate it.
Appreciate it.
Stay safe, guys. Thanks.